Hi, Dr. Newmark.
Hi,
Dr.
Darby.
Newmark is a chair in regenerative biology at the Morgridge Institute for Research. He received his PhD from the University of Colorado Boulder. Interestingly enough, he is a very talented trumpet player, which was exciting to learn. He actually almost pursued a career or a degree in music rather than science, which wouldn't have brought him with us here today. So the Newmark Melt Lab, which he's going to talk to us about, uses both wet lab and a computational approach to study methods of regeneration. And specifically, he uses free-living flatworms called Planarians that he's going to talk to us about today.
Okay, hi everybody. It's really a pleasure for me to be here. I wish I were there in person because I think, as many of you know, I'm an alumnus of this department. It really was played a huge part in the kind of science I do and the way I approach science. And I'm just sad I'm not there to be able to see so many friends and colleagues in person. All right, so what I'm going to do today is talk to you about some of the work from the lab. And actually, I'm not going to focus on the planarian work today. The invitation was to talk about these creatures, parasitic flatworms known as schistosomes. So what I'll tell you about today is how our interest in developmental biology and studying the developmental biology of these parasites has led us to some interesting observations about the ways in which these parasites might actually be evading their host immune system. All right, so this work actually has its foundations at Carnegie. So as many of you know, if you take a freshwater planarian, this free-living flatworm, and lop off its head, within about a week or so, it will grow back all the missing tissue. And in fact, any part of the worm can regenerate a complete animal. And they can do these, they can regenerate so well because of a population of stem cells that these adult planarians maintain throughout their adult lives. In these animals, the only proliferating somatic cells are these stem cells known as neoblasts. So what you're looking at here is an animal that's been labeled to detect the proliferating cells. You can see there's lots and lots of them in these worms. And really beautiful work from Peter Radine's laboratory has shown that within this heterogeneous population of stem cells are true pluripotent stem cells, because transplantation of single cells into lethally irradiated worms can restore all cell types viability and lead to generation of all the missing tissues. So in my lab, we've been interested in understanding how it is that planarians, in addition to being able to do interesting things like make a new head and a new brain, we're actually intrigued by the fact that they can make an entirely new germline from parts of the animal that are missing reproductive organs. And that's what the planarian half of the lab studies. And I'm not going to talk to you about that today, but hope to maybe see you guys in person at some point and tell you about our planarian work. Now, as we were studying planarians, we began to see many parallels with their evil cousins, parasitic flatworms. What you're looking at here are a male and female pair of Schistosome mansoni. These schistosomes are responsible for the disease schistosomiasis. And so you have a sense of the scale of this problem. This is one of the major neglected tropical diseases on the planet. Over 200 million people are infected worldwide. Okay, so this is a huge, huge problem in the developing world. So what you're looking at here, this guy up at the top, this is the male of the pair. This is his mouth. So these are blood flukes. They live in our veins and they eat our blood. This is his mouth that he uses for ingesting blood. This is the ventral sucker that he uses to attach to our vasculature. And then in a groove along the ventral surface is his female partner, and they remain paired throughout their long lives. Now, it turns out that the pathology of the disease is not due to having these worms crawling around in our veins, which is gross, but as I'll tell you, these parasites are quite good at disguising themselves from the immune system. And in fact, the pathology is due to the fact that these females are highly prolific, and the eggs that they lay are highly inflammatory. And so those eggs can actually embed in host tissues, triggering massive inflammatory reactions that lead to the chronic tissue damage associated with the disease. All right, so these females can lay hundreds to thousands of eggs per day, depending upon the species. And as I'll tell you, the adult parasites can live for years. Now, right now, there's just a single drug that's used to treat schistosomiasis. It's pretty effective, and it's used in mass drug administration programs. So there are serious concerns about resistant strains emerging. We'd like to have other ways of targeting these parasites. And so we'd like to know a lot more about their biology. Now, what I'm going to tell you is these are obligate parasites of 2 different organisms. And that complex life cycle has made studying these worms quite challenging. And so when Jim Collins was a postdoc in the lab, his work, and actually work that I had done, that Alejandro and I had done back at Carnegie together, had already pointed us in the direction of trying to take parallels in some of the shared biology between planarians and their parasitic cousins, and now using them to understand those parasites. And this is work that Jim pursued in my lab and now in his own lab at UT Southwestern. So I need to walk you through this life cycle. And hopefully we can appreciate this not as a parasitology 101 torture session, but as a really fascinating example of developmental biology. So I talked to you about these adults living in the mammalian vasculature and that the females lay lots of eggs. Based on blood flow, a lot of these eggs wind up embedding in host organs, causing tissue damage. Eggs that make it out of the host and hit fresh water will hatch out this little creature known as a Mini-Symposium. It actually looks like one of Laura's protozoans, not quite like, not as beautiful as Oxytricha actually, but it does have the ciliated outer surface, but this is a multicellular creature, about 150 microns long. And in its 24-hour or so lifespan, it has one job, which is to find the appropriate species of snail intermediate host. If it finds that snail, it does something remarkable. It will invade the snail and then shed most of its differentiated tissues and convert itself into this stage called the mother sporocyst. I apologize for these names. I did not come up with any of them. But basically, the way to think about the mother sporocyst is that it's a Russian doll of bags of stem cells. So within this mother, bags of stem cells develop. Those are called daughter sporocysts. The daughter sporocysts will leave the site of infection, migrate deeper into the snail tissue where they become associated with the snail organs. And those daughter sporocysts can then reproduce themselves indefinitely. So one can just serially transplant daughter sporocysts from infected to naive snails until one gets tired of doing the experiment. So these can continue to replicate, or they can do something truly remarkable, which is without fertilization, they can enter embryogenesis directly to give rise to the next stage of the life cycle called the cercaria. So what is shed into the water from infected snails is the cercaria. This is what they look like as they're swimming. This is a high-speed video. So that's what it looks like to the naked eye. And then if we slow it down, what you can see is that the parasite is using its forked tail to, like oars, to propel itself backward through the water. And it can actually go forward or backward depending upon how it deploys some muscles. So these parasites are shed into the water column where they search out a mammalian host. And if they find such a host, they'll penetrate the skin in a matter of minutes, shed their tail on the way through, and convert to this larval stage known as a schistosomulum. This schistosomulum then has to work its way from the skin to the vasculature, to the lungs, and ultimately to the hepatic portal vein, where the males and females will find each other as juvenile parasites. And then this pairing is required actually for female growth and reproductive maturation. Okay, so within this complex life cycle is really fantastic and fascinating developmental biology. And we're intrigued by how this single parasitic genome, which after all has lost many genes because these are obligate parasites, how this pared down parasitic genome can specify 5 different body plans depending upon the environment in which the organism finds itself in. All right. Now, when Jim started this work, he was intrigued by this longevity that I alluded to earlier. So people can leave endemic areas and still be shedding eggs, so meaning that they have reproductively mature parasites inside of them for decades. And in fact, as much as over 30 years, people have left endemic areas. And you cannot be self-infected. You can only be infected by this stage that comes out of the snail. So that means these adults can live for decades in the host. And Jim was curious about how it is that these parasites could live for so long in the inhospitable environment of the host bloodstream. So he hypothesized that they must have robust mechanisms for maintaining their tissues. And because these are parasitic cousins of planarians that use their stem cells to regenerate and actually for tissue homeostasis, Jim hypothesized that these parasites might also have similar kinds of cells. Jim went on to show that that is the case, that the parasites do have neoblast-like cells. For our purposes today, what we need to know is that Jim showed that these cells express paralog, one of 2 paralogs of the germline regulator nanos. This particular one is expressed in the somatic stem cells as well as the germline. And Jim also showed that maintenance of these cells requires an FGF receptor related molecule. Jim then went on to show that one of the major functions of this population in the adult parasite is to maintain this outer tegument. So this is this amazing double lipid bilayer, a syncytium that surrounds the entire parasite. And the stem cells are generating a huge number of precursors for the tegument to maintain it in the context of this presumed battle between the parasite and the host, where this structure is playing an important role in, and actually is the surface with which the parasite interacts with the host. Now, with the demonstration that there were such cells in the adult parasites, we wondered if there were if these kinds of cells were playing other roles in the life cycle. And at this point, I was fortunate to have Bo Wong join the lab. So Bo actually doesn't normally have a rainbow colored afro, but when I asked for a photo of him, this is what he sent. So I get to use this for the rest of his career, which I'm enjoying doing. But Bo came to the lab with a background actually with a PhD in material sciences and engineering, and became interested in developmental biology and joined my lab. And he was intrigued by the fact that a single one of these Miracidia, as I told you, can infect a snail and then essentially generate through asexual reproduction a continuous immortal supply of daughter sporocysts and ultimately these infective cercaria. So Bo then went on to show that in fact there were similar cells that he could identify that were packed into the Miracidia and then passed into the molluscan host, and these cells then are amplified and drive this asexual reproduction inside the snail. Now, Bo's work showed that there were that this population of stem cells was heterogeneous. He went on to do single cell sequencing, and what he found was that there was a population of cells that expressed the two genes that I told you about in the adult stem cells, this nanos gene, nanos2, as well as this FGF receptor. He found a separate population that expressed only nanos and not the FGF receptor. And he found a third population that expressed the FGF receptor and not nanos. Okay, and so we wondered what these cells were doing in the rest of the life cycle. And so what you're looking at here is a fluorescence in situ hybridization image from a cryosection of an infected snail. And what you're looking at here, this is the cercarial embryo developing in the snail. So this is the body that I showed you. The tail, the muscular tail that does the swimming would be off the screen at the bottom down here. Okay, so this is now in situ hybridization, detecting nanos2 in green and FGF receptor in magenta, and then nuclear labeled blue with DAPI, and then the penetration glands that the parasite uses for secreting digestive enzymes from the anterior are labeled with a fluorescent lectin. So what Bo saw was this really intriguing pattern where there was a pair of these double positive cells on each side of the parasite on the left and right, as well as a cell at the midline, and then clusters of nanos positive cells in the posterior of the cercaria. Now, to ask what happens upon infection of the mammal, that is a challenging question. But so, and let me tell you a little bit more about the biology. So what you need to know is that, so what you're looking at here is just a growth curve with the morphology of the parasites amplified. This is from a classic paper from Clegg from 1965. Average length of the parasite in millimeters is shown on the left. And so what you can see is as the cercaria migrates to the lungs a week later and ultimately to the hepatic portal vein 2 weeks later, what you can see is the parasite's growth really doesn't, the parasite really doesn't grow in this 2 week period, but it is changing dramatically. And it actually has to take a gut primordium, this upside down Y here, and turn it into a functional gut, such that when the parasite reaches the hepatic portal vein, it can now ingest blood, and that blood will then feeding will then fuel the exponential growth of the parasite. Okay, so because there isn't any growth in this initial 2 week period, and because we had only recently identified the stem cells, no one had looked carefully at what was going on in terms of the proliferation and what cells were proliferating and what was going on developmentally during this early time point. So the earliest identified mitotic figure actually was at 4 days post this transformation. So at this point, I was really fortunate to have Jae-Hun Lee join the lab as a postdoc. So Jay's background is in epidermal stem cell biology. And what Jay was able to do now is to take a skin biopsy from a mouse, overlay it on appropriate media that had been developed, in this case by Paul Bosch, who was a former really fantastic schistosome researcher at Stanford. And what Jay can then do is take cercaria from snails, add them to the skin in this ex vivo explant, the cercaria will then infect the skin, penetrate, swim into this media, shed their tails on the way through, and they'll convert to this intramammalian stage known as the schistosomula. Okay, so let's just take a look at what this looks like. So what Jay's done here is labeled the skin in DAPI, and then the parasite is green fluorescent labeled with DIO. And what you can see here in the spinning disc confocal image is the parasite interacting with the skin and penetrating. And we'll play that one more time. So you can see here the parasite working its way through and then going through the skin. Those of you that looked carefully saw that the parasite actually took its tail with it. That's because this guy cheated. It went through a broken hair follicle here. So typically they would not go through a broken hair follicle. They'd have to work a little bit harder and their tail would be shed on the way through. Okay, so coming back to this assay now, if we want to ask what happens with respect to cell proliferation following this transformation, we can ask, do cells proliferate, and if so, which cells are they? Jay can now add EDU to this culture media and now basically monitor the incorporation of this nucleotide analog during the S phase. And Jay saw something truly remarkable when he did this. So if you look within the first day and a half, what Jay found was reproducibly this handful of cells, hair on either side, and this cell at the midline, that were entering S phase and were beginning to replicate. If you carried this out a little bit longer, you could actually see these cells replicate and basically double. And interestingly, if the pulse was done later after transformation, in this case 9 to 10 days post-transformation, what Jay saw was another population at the posterior that had begun to proliferate. Okay, so from these EDU labeling experiments, oh, and what I should tell you is that these cells, it's no coincidence that we saw the same pattern, because by double fluorescence in situ combined with EDU, we can show that these EDU positive cells are the ones that express both nanos and FGF receptor. Okay, so it looks like these are the first cells to proliferate upon entry into the mammalian host, and we saw that there were early and late proliferating cells. All right, so to summarize what I've told you so far, essentially the work from the intra-snail stage of development identified this heterogeneous population of stem cells. This population that does not express nanos turns out to be behaving like a transit amplifying cell that is associated with transitory larval structures like the tail. We believe that this double positive population gives rise to soma and cercaria, but then is also packaged into the infective form such that it can be passaged to the parasite, passaged into the mammalian host. And these double positive cells, we believe, are the first to enter the cell cycle, and that these give rise to the somatic stem cells in the adult parasite. Now, this other population that only expresses nanos, from Bo's earlier work, these were slower cycling cells. And what we think is happening is that, again, these cells are also packaged into the cercaria and transmitted into the mammalian host. These cells, through transcriptional profiling, Bo identified a novel schistosome-specific gene that we called LED that marks this new population upon entry into the mammalian host. And these cells wind up basically contributing to the germline, as well as this posterior growth zone. So that I didn't emphasize this enough, but the growth that the parasite undergoes after reaching its mammalian niche is basically allometric. It's a massive posterior growth, and there appears to be a transitory population of stem cells that are derived from these LED cells that contribute to that. All right. Now, we wanted to dive in more to look at what was happening in these earliest stages of parasite development inside the mammalian host. And I'm going to talk about the relevant organs for what's happening early on as the parasite develops. So the first thing we'll talk about is this parasite's outer surface tegument. Okay. Now, as I told you when Jim was in the lab and now in his own lab at UT Southwestern, he went on to show that the stem cells will give rise to tegumental precursors that express the transcript for a tetraspanin, the protein of which is expressed on the surface tegument. Now, this tegument is a fascinating structure. As I alluded to, it's a double lipid bilayer. It's built in the initial 2.5 to 3 hours post-infection. What the parasite does after invading the skin is it sheds its outer glycocalyx, And then it builds de novo this novel double lipid bilayer. Okay, and it's thought that that second bilayer is involved in cloaking the parasite from the host by taking up, for example, host factors and displaying them. All right, so we're in this case, so we know developmentally that one of the first things the parasite does is build its tegument, and we're going to use this gene TSP2 to mark tegumental precursors. And the image you're looking at here is just showing you that the cell bodies of the tegument are actually buried underneath the body wall musculature, which is here in magenta. The surface tegument is out here, and then there are cytons that connect the nuclei of this syncytial tissue out to the outer surface. So synthetic materials are provided to the outer layer via these processes. All right, so the other thing the parasite has to do, of course, as I told you, is it needs to make a gut and digestive system. So one of the other structures we'll talk about is the esophageal gland, shown here. And I will tell you why that's important. We have the marker we're going to use to detect it is a microexon gene that is expressed abundantly within this structure. And then there's the gut proper, and we're going to use cathepsin B to follow the development of the gut branches in these parasites. All right. So if we look at these tegumental progenitors that I mentioned, this is this is fluorescence in situ hybridization of the infective cercaria. And what Jay sees is that there's roughly 30 or so of these cells that are found in a cercaria. And now if we undertake the parasite through this in vitro transformation, what Jay sees is that there's a decrease in these tegumental precursors, presumably as they're being incorporated now, as these precursors mature and become incorporated into the parasite's outer surface. So we see a significant drop over the first week and a half of parasite development. Again, this is in vitro transformation. To confirm this is real, Jay's harvested parasites from the lung stage and shown that there's a similar drop from the original infective larva to the parasites that are recovered from the mammalian lung. Okay, so these precursors are gradually depleted. And now we wanted to ask if the stem cells were also forming new precursors. And so what we can do again is transform these cercaria to schistosomula in vitro. In this case, this can be done by also mechanically shearing the tail and then subjecting them to an EDU pulse, and then either fixing them immediately or chasing them for an additional several days. So this is what this looks like. So if, as I showed you before, if Jay does a pulse in the initial 3 days, he sees this handful of EDU positive cells in turquoise here, and none of the TSP2 labeled cells, which are magenta, are labeled at this early stage. However, if he now chases the EDU for several days, he can now detect EDU positive cells that express this tegumental marker. And this is just quantified here, showing this increase. This is a slightly different developmental period. This is now a 2-day chase after a 4-day pulse, but the point is the same, that these double positive cells are now found and increased dramatically. So these early proliferating cells are giving rise to tegument at this very early stage of transformation. Now we wanted to ask what was happening with the digestive system. So what Jay has done here now is harvest lung stage parasites from mice that had been infected and then given an IP injection of EDU. So this will actually label the parasites in the mice, and now we can harvest them over a time course of various chase lengths. So again, looking at the cercaria, what you see here is that this infective larva has a primordium for the esophageal gland, which expresses this gene MEG4, and the cercaria has a primordial gut expressing this gut marker cathepsin B. Now, if Jay harvests parasites after performing such an EDU pulse on the host, this is now taken from the lungs at day 8 of infection, what Jay is able to find is that about half the parasites he recovers actually show EDU incorporation into cells expressing the esophageal gland marker. So these cells have chased into the esophageal gland at the lung stage. Interestingly, when Jay does the same experiment with the gut marker, he never sees incorporation in the gut at this lung stage. This was really surprising to us because one would imagine that the gut and its associated organs, in this case this gland associated with the esophagus, that these would form together. And that wasn't the case. We see the esophageal gland forming earlier than the gut proper. All right. Now, in the interest of time, I'm just going to kind of summarize a lot of work just to give you a sense of what's happening here. So as I told you, this infective larva that penetrates the mammalian skin has a handful of stem cells, colored yellow here, packaged into it, as well as primordia for the esophageal gland and gut, and then precursors for the tegument. And what I've shown you is that in this initial stage, as the parasites, after penetration, as the parasite's working its way from the skin into the vasculature, the stem cells are basically giving rise to tegument. We don't see them making other lineages. And in fact, the number of stem cells is not increasing during these very early stages. So the stem cells seem to be dividing asymmetrically, giving rise to tegumental precursors, and then making an additional stem cell at each division. If we now harvest parasites from the lung stage, we can see that we can still chase cells into the tegument, but now we can chase cells into the esophageal gland. And again, the number of stem cells is still constant a week after infection. But now once we harvest parasites from the hepatic portal vein where they can initiate eating, now we can begin to see cells giving rise to gut tissue as well as making additional stem cells. And so what this tells us is that there's this period between migration from the lungs to winding up at the hepatic portal vein where the gut is actually being formed. That is currently experimentally a difficult area for us to get at right now, but that's something of very much of interest to us. And then what happens is now that these parasites have built this gut, they're able to eat and continue growing. And then this growth is fueled by this second population that I alluded to earlier that will give rise to these cells of the posterior growth zone. All right, so with this sort of summary of some of these really important developmental events happening during intramammalian development, we wanted to know if we could find regulators of these processes of early development, if those would tell us anything interesting about the parasite. So what Jay did was he looked for transcriptionally at genes that would be upregulated in the early intramammalian stages. And one of the genes he identified was a schistosome homologue of the conserved endodermal regulator FoxA. So this is well known for its role in organ development in the pharynx and C. elegans, the pharynx in Planarians, and endoderm development in mammals. And in schistosomes, it's expressed abundantly within the esophageal gland. This is a fluorescence in situ image with FOX expression shown in yellow, and then this esophageal gland marker in magenta. This animal's anterior is here. Here's the ventral sucker. The mouth would be out here. So the gland, again, surrounds the base of the esophagus. Okay, so we wanted to know what FoxA was doing in the parasites. They're susceptible to RNA interference. So we can harvest parasites from a mouse host, and we can do this various time points post-infection. So we can harvest juveniles, we can harvest adults, soak them in double-stranded RNA in the appropriate culture media, and fix them after a couple weeks in vitro. And this is what control parasites look like. So again, here's our esophageal gland marker in the control. If it's, of course, forming very nicely in vitro. Here's the gut marker, and you can see the morphology of the gut branches that I've shown you earlier. And now, when Jay looks at the FoxA knockdown parasites, he sees something quite dramatic. That is, the gut forms normally, but the esophageal gland is completely missing. So the structure is not there. We've used several markers to validate that. Now, and what I should say is we don't see differences in the growth or the ability of these parasites to eat in vitro, so they can still ingest red blood cells, which we can include in the media. They can convert those red blood cells, turn the toxic heme into the hemozoan pigment that detoxifies it, and so their digestion seems to be okay. And so this organ seems to be dispensable for survival or growth in vitro. Now, what this reflected to us was this was not telling us that this organ was not important. Rather, it told us that this is a problem with studying an obligate parasite in vitro. They're basically in this beautiful spa of culture media that gives them everything they need. They're not being subjected to the host immune system and all the other nasty things that are happening inside the vasculature. So we thought that a different assay was needed. And so at this point, we needed to move in vivo. And these experiments are incredibly challenging. When Jim was in the lab, he was fortunate to go to Donato Cioli's lab in Rome. And Donato had worked out these transplant procedures for taking parasites from one mouse and now surgically transplanting them into another mouse, or hamsters in his case, and then mice. So Jim had developed these techniques and then was kind enough to teach Tracy Chong, a senior scientist in the lab, who's incredibly skilled. This is a wedding cake she made for one of the lab members. And her artistic skills come in handy for these very difficult surgeries. So what she and Jay did was to take these adult parasites, subject them to FoxA double-stranded RNA, and now transplant them into naive mice. And what you can see here is that for control parasites subjected to control double-stranded RNA, basically you can recover roughly 60 to 70% of the worms that you introduce. By contrast, we can barely recover any FoxA RNA parasites. So this said that this gene is in fact required, and this gland is required for survival in the host. And if we look now at the livers of the target mice, what you can see here is, here's a liver from a mouse that's been treated with, or in fact, has had control parasites introduced. And what you can see is all these tiny white dots all over the liver. I trust that you can all see that. Though each one of those dots is a granuloma that is formed where an egg is embedded. By contrast, the FoxA RNA livers look quite good. They're quite clear, except for these giant splotches around the outside. And in Jim's initial work describing this technique, it turns out these large white blobs are dead worms that have been flushed to the periphery of the liver. Okay, so these worms are not viable. Now, one might be thinking, well, this is just a trivial result. Basically, this is a nutritional defect. The animals don't have this digestive organ, they're not eating. And that's a completely reasonable hypothesis, but Jay did a really cool experiment. So again, here's transplants into wild type mice, showing that you can barely recover FoxA knockdown parasites. There was really lovely histological work from Alan Wilson's lab at York that suggested that the esophageal gland could actually trap host immune cells. And so Jay thought, well, what would happen if we actually remove the immune cells from the mice? And so what you're looking at here is now transplantation into rag knockout mice that are lacking both B and T cells. And what I hope you can see is that now, under this condition, that rescues the lethality of these FoxA knockdown parasites. And to hone in on this even more, Jay went on to show that in UMT mice, which lack B cells, lethality is also reversed. Okay, so in fact, B cells are required to kill these FoxA knockdown parasites, the parasites that lack the esophageal gland. And so to give you a sense of what we think is happening here, if we now take parasites in vitro and feed them GFP labeled immune cells, what you can see is they can take up the cell. This is the mouth. The immune cells now are passed to the esophagus, pass through the esophagus, where they wind up at the esophageal gland, which is at the very base of the esophagus into the gut. And what happens then is the worms gulp and all fluorescence is gone. Now, intriguingly, when Jay does this with the knockdown parasites, they eat just fine. The cells are taken up just fine. They accumulate at the base of the esophagus and the gut just fine. But now when they swallow, the cells are passed directly into the gut. So this barrier is completely lost. All right. So this is a complicated model. I just want to give you a sense of how we're thinking about this. In these control parasites with the esophageal gland, we believe this is acting as a barrier to host immune cells, but it's allowing red blood cells in. Because we can see them being taken up during the feeding process, and we can see them being converted to hemozoin, and we can see that the parasites then regurgitate this along with host molecules. And this is what we think is leading to adaptive immunity to internal antigens from the parasite. And what we think is happening is that in the absence of this esophageal gland now, antibodies and various immune effector cells now can access these internal organs and actually digest the parasite from the inside out. So what I've told you today is that by looking at the developmental biology of these parasites, we found the subset of stem cells that's passed from one life cycle stage to the next, and that these cells are driving the development and growth of the intramammalian stage of this parasite. And then that by looking at this one important developmental regulator, that told us something really interesting and unexpected about the role of this esophageal gland in evading the host adaptive immune response. So with that, these are the folks I need to thank. This is my current group. As Laura alluded to, it's really hard to be apart from the lab for this long. Missing them all. They're a fantastic group of people. Just another shout out to Jim for developing the schistosome work. He's now doing great at his own lab at UT Southwestern. Bo for the fantastic work on intramolluscan development, who now has his own lab at Stanford. I talked about Jay and Tracy. They got help on the life cycle maintenance from Jaron Gao and Jamesh Patel, the rest of the group. And I want to also thank Fred Lewis of the Schistosome Resource Center, who welcomed Jim and me into his lab there to teach us how to work on schistosomes, and I will be happy to take any questions.
Thanks so much, Phil. Great talk.
Thank you.
So your first question from Yishen Zhang, we have, how can the barrier block immune cells but not other cells?
Yishen, that's a great question. I have absolutely no idea. So I'm giving you our last experiment up until the shutdown. So all I can tell you is that we know that the red blood cells are getting through because we see them being digested properly. But exactly how this is working and what this barrier is doing, we do not yet understand. So what we want to do now is basically sort of look closely at that barrier with fluorescently labeled red blood cells, with different fluorescently labeled B cells and other immune cells, and basically try to examine what's going on there and to ask whether this barrier is also affecting the ability of antibodies to access and those kinds of questions. So we don't know. It's a great question.
Awesome. Did you see Yishen's response? She said, perhaps surface proteins.
So I should have said a little more about that. So these microexine genes so that's the idea, that likely there's something that's secreted into the lumen of the esophagus by this gland. And there are many, many specific factors that are secreted by this esophageal gland into that lumen area. And basically from the work of Alan Wilson, what he showed was that these immune cells are in fact, basically what his observation was that you could see immune cells in the lumen at the esophageal gland, and you never see immune cells inside the gut. And that's what led Jay to do these experiments in the first place. So the idea is that there's something about the proteins that are being produced and secreted into that part of the lumen of the esophagus that are trapping those cells. But how they're doing that specifically, there's going to be work for the future.